Hello, everyone. First of all, just like you said, I really can't thank enough to the program as well as amazing mentor, Dr. Julie Cheriton, and who has really taught me so much what I know now about this whole new field to me. So, and also thank you for listening. And my name is Huang. Well, I guess that's done already. I'm a faculty at Northwestern Fabric School of Medicine. Our goal is trying to develop anti-cancer drug through an alternative approach from the conventional one. And through that approach, today I present to you a new chemical entity, Ming. Ming show efficacy against human liver cancer, both in vitro and in vivo. In a head-to-head direct comparison analysis with the current standard care, sorafenib, Ming shows a superior efficacy in HCC xenograft modeling. Ming also has a multi-targeting mode of action and is also not a substrate of activated T-glycoprotein, renders it less likely to induce drug resistance. It shows a good tolerability in vivo. Northwestern holds IP towards the drug, and we aim to advance the compound towards preclinical, ultimate clinical testing for, in this case, HCC, liver cancer indications. Our team has prior experience that forward another lead compound from the same developmental program. Now it's currently in clinical trial, transitioning into phase 2 at NCI. So as you all know, that cancer is a complex and heterogeneous problem. And to this day, factor or cellular pathway that are necessary and sufficient for cancer development remain unknown, making it difficult to identify specific drugs for selective inhibition against cancer. That's part of the reason that cancer remained number 2 killer in this country, before the COVID started. And current target therapy that targeting single gene and gene products have been in clinic for more than 10 years now. Although it showed efficacy in the early stage, often they targeting very specific tumor cells. For example, cells bearing targets for those specific drugs. When those cells are removed, however, remaining cell generally take over and causes tumor relapse. Our goal is trying to look for strategy to target cancer cell unique property that's shared by a lot of other cancer cells, hoping to lead to a more broader impact on tumor cell populations. To do so, we have developed an in vitro assay to hope to reduce the complex in vivo malignant phenotype into an in vitro platform for us to identify small molecular hits. And in so doing, we identify a cellular structure. In fact, it's a nuclear body called perinuclear compartment. And this structure is a multi-component structure containing primary RNA, RNA binding protein, as well as DNA structures on it, and only present in cancer cell and is absent in normal cells, even including pluripotent ES cells. And the presence of this markers in cancer cell can be scored as a PNC prevalence. That means the percentage of cell containing at least one of this structure. And what we found through multiple in vivo and in vitro model, the percentage of cell containing at least one PNC increases as cancer progression, and it's inversely correlated with patient survival. Therefore, it's suitable to be used as the phenotypic marker that marks the malignant behavior of cancer cells. Using phenotypic marker for small molecule screening provides a couple of advantages. One, the uniqueness of this marker allows identification of hits that potentially target multiple functional relevant cellular protein and factors that are perhaps required for the tumor growth and tumor expansion. And secondly, the uniqueness of the structure in cancer cell allows the identification of molecular with less toxicity against normal cells that don't have the structure. With this as a high content push-screen marker assay, we have identified already a lead compound, meterestin, which is currently in clinical trial going into phase 2 at NCI. Distinct from Northwestern, Ming show a particular suitability for liver cancer indication. Ming has been proven to have an inhibitory effect against multi-oncogenic drivers. As you can see that in tumor, xenograft tumor, in this case, HUH7, it's a liver cancer tumor xenograft. The treatment of Ming significantly represses at 15 mg per kg, the oncogenic protein SIR1 as well as MYC. In comparison, sorafenib does a much poorer job. And in addition, Ming substantially increased the expression of tumor suppressors, as you can see here. Another tumor suppressor that are significantly increased by Ming is the microRNA 141 and 200, whose expression is substantially reduced in tumor cells compared to the normal liver cells next to it. However, the expression is restored when the treatment of Ming commences, as you can see here. And most appropriate for liver cancer, particularly in this country, is Ming is significantly inhibit a profibrotic gene expression. That's a connective tissue growth factor, which plays a key role in liver cirrhosis, which has been increasing incidence in the United States due to a prior epidemic of Hep C, as well as the lifestyle issues. So at RNA level, the treatment of Ming can substantially reduce CTGF by more than 6 log-fold, making it particularly a good candidate for liver cancer treatment. In vivo, Ming showed efficacy against multiple xenograft models, HCC xenograft model. As example, this is a HUH7 xenograft model. You see treatment Ming at 15 mg per kg significantly block the growth of the xenograft tumor. And in comparison, same dosing, sorafenib does a much poor job. And double concentration of this 15 mg per kg at 34 mg per kg would induce a tumor regression so 19 to 49 days with very little impact on body weights of treated animals. Preliminary tox analysis with a double concentration of Ming's effective dosing is 75 mg per kg single dose injection do not find any abnormality as far as body weight, organ weights, organ physiology, histology, as well as blood panel in 48 hours post single administrations. Therefore, we feel like our finding about Ming make it particularly suitable candidate for further investigation to be a drug candidate for liver cancer. As liver cancer now become a prominent problem around the globe, in US, the incidence is being increased due to Hep C pandemic and the lifestyle issues, and patient has very little options. The only curable strategy is liver transplant, and which is far few and in between. Most patients, standard care is being the sorafenib and its derivative, and that is shown to be limited effective with substantial side effects. And Ming having a different mode of action from the kinase inhibitor, and could provide a new opportunity for option for patients, also can be used as a synergized compound with the existing standard care. And our plan is to advance Ming towards pre-IND and IND enabling work, ultimately clinical stage, in addition to explore other indications, for example, gastric cancer, where we have in vivo data show tumor regression, as well as renal. Furthermore, we are developing pro-drug that will increase selectivity against cancer cell in multiple indications as well. And the team is composed the Northwestern group, as well as Dimitra, who is with a strong chemistry background, as well as pharmaceutical experiences. And in summary, Ming is a novel small molecule that show efficacy against liver cancer, both in vitro and in vivo. And it's in a direct head-to-head comparison analysis, it shows improved efficacy against HCC xenograft than the standard care sorafenib. It has a multi-targeting mode of action that inhibits more than one oncodrivers and also activates multiple tumor suppressors. Most of all, unique for liver cancer, it inhibits the profibrotic factor CTGF, and which is the key player in cirrhosis of liver, and that often precedes to liver cancer. Furthermore, MING is a very well tolerated compound in a long-term treatment in mice, and it's also not a substrate for activated P-glycoprotein. So we're seeking for initial $1 million to do groundwork for IND enabling studies, including formulation work to achieve maximum oral availability, PK metabolism, as well as in vitro and in vivo toxicity evaluations. Last but not least at all, I would like to thank my amazing mentor. I have to say, she just taught me so much, and Julie Sheridan, and the Equalize 2022 program that gives women like me so much support and education opportunity. And last but not least at all is the NU intellectual property managers, Dr. Michael Moore, who have moved on and still helping me, and currently Ivana, I'm sorry, I didn't say it right. Most of all, I'd like to thank you all for helping, for listening, and I love to hear the comments from you guys.
Great, thank you so much, Sui. The first question/comment will come from Chris Borner.
Hi, thank you so much for the presentation. Very interesting. Obviously, these cancers are areas of high unmet need, but they're also been quite dynamic of late. And given that immuno-oncology agents have increasingly displaced sorafenib in some of the markets that you're looking at, notably HCC and gastric, and I noticed renal was on one of your slides. Have you looked at the synergies that this agent would have with prevalent IO agents, or have you taken a look at IO experienced models as you evaluated the profile of this potential target?
Yes, that's a great question. In fact, immunotherapy is coming up to be one of the choices in liver cancer patients, but however, it has a limit to a really subpopulation. So still have a lot of people are not compatible for that treatment strategy. And we are intent to work with combination of therapy with the immunotherapy reagents in syngeneic mice, and to see whether that can really enhance the efficacy in those circumstances. That's a great question. Thank you.
Thanks, Chris. Chris Garabedian.
Yeah, sure. First, I wanted to share your esteem for your mentor, Julie Charrington. I worked with over 20 years ago at Gilead, so I know she gives great advice. So help me square the circle a little bit on the phenotypic screening to identify the right patient population and the very compelling sorafenib mouse model. It looks like those error bars are very tight and that you must have had a lot of subjects, but was there specific criteria that went into those PDX mice and selection of that? And if not, right, just help me understand the strategy to enrich the population in a clinical study and what you learned from that xenograft model.
Yes, it's a very good point. We're actually trying to work out exactly what sort of targets are involving through the drugs that come from the phenotypic screening. In fact, it's multi-targeted, like as I showed, like there's already 2 of them. So I hope I answer your question correctly, and please let me know if I don't. So we will try to develop molecular profile for those that are sensitive to for MING. And so by so doing, we can stratify patient population to see whether the tumor do have components that are changeable by what MING is targeting.
Yeah, I think elucidating that trade-off, right, to get to a more enriched population versus a broader population and what that looks like, I think is important. The other thing is $1 million seems pretty light for your IND enabling and other work, formulation work that you wanted to do. So you don't have to comment on that now, but that may raise questions for some investors.
You're absolutely right. Yeah.
Okay.
Great. Thanks. Meghana.
Yeah, I was just going to dig in a little bit to the, what your sort of plan is for the money, the $1 million. I sort of had a kind of similar reaction. But yeah, again, you don't need to comment on it. And I think just maybe fleshing that plan out a little bit more, it would be helpful in the future.
Yes, we actually have a clear plan because we wanted to do a non-GMP study first to get a handle before we ask more to, this is really a pre-IND enabling work. And so trying to get an idea, the basic PK and the tox, as well as the formulation to get the maximum oral absorption. That's actually the first thing to do. So we already get quote from the CIO to get those work to do, this 3 pieces of work to be done within a few months of the time. Is about $1 million. But based on those work, we can expand to like scale up in a GMP work for tox.